What is up? It is good to see you guys. This should be theoretically Monday. Um, Hopefully, I'm not dead? Question mark. Um, we are going to spend this week, and of course, I've gone over this with you on Friday to kind of give you an idea of what's up. But we're going to spend this week doing the ideas of active transport, finishing some passive transport, but really active transport, and then prepping you for a test. Legit, all we're doing. Okay. So we have this lecture today. We have tomorrow is going to be my review on active and passive transport. Wednesday, because I was worried about some of the dance cheer being gone, you've got some book work for me to do. And then on Thursday is your test. So let's make sure that we understand something. I'm not here. You don't get to ask questions of me, whatever you want to say. And yet, I'm going to be giving you all the information that you have to have right here. I gave it to you when I lectured on passive transport. I'm going to give it to you when I lecture on active transport. And you're going to see it in a review. And this video, if I can make it happen, will be on RenWeb. And so you can watch it 4,000 times for all I care. My point is that we don't have an excuse. Again, we have got 3 grades to start off this 9 weeks. 3. Of those 3 grades, one of them was a give me. I gave you a free grade. It wasn't free for moms and dads that had to pay for all the parts. I got you. But I gave you a free grade of us working on our sales. Okay? Should have been no question. By the way, I may as well start my stopwatch so that I don't talk too long and keep y'all here into the next class. But I gave you that information. So there should not have been any questions about that side of it. All right? The second thing is that we took a test right before I left on the sale. And to be honest, it should have been the easiest test you took. Lastly, you have the ability now to take another test. While, yes, I'll agree it's on active and passive transport, it's not something that we typically spend a lot of time in 7th grade or below learning. It's still not complicated stuff. It's not photosynthesis. It's not cellular respiration. It's not DNA and transcription and translation. Like, trust me, we headed to far higher mountains than where we are right now. But what I mean by that is that start your grade off right. Like, get a head start. Don't put it off. Don't be, I got to fix the problem come the end of the semester. Don't do that. Help yourself out. Okay. Do want to make sure that we understand that there will be a quiz on Wednesday. It is your Latin terms 6. Okay. I will have already given those out to you because I'm thinking about that today on Wednesday before I leave. So I will make sure that those are in people's hands probably Wednesday, Thursday, Friday. I'll have them available for anybody that needs to get them so that you have your Latin terms for 6. So that will be on that Wednesday, and then you'll still take a test for me on Thursday. Please feel free to realize there may be a pop quiz while I'm gone. Again, look, y'all, it's not that I dislike my 9th graders. As a matter of fact, you're a better class than what was given to me as far as statements about the 8th grade class last year. That was y'all, if you didn't know. I really do enjoy every single one of you. I do. But I want you to understand nothing we do that's worth anything is easy. It's not. I'm fully aware there's some of you that will never go into biology. I'm aware that some of you will never do medicine. I can respect that. What I can't respect is that because something's difficult, we just give up on it. Don't be that person. I'm not trying to be judgmental, but if you give up on this, you're the same type of person who gives up on a ball team. You're the same type of person who gives up on a relationship that's difficult. Boyfriend, girlfriend. It's not really working out. I just don't want to fight for it. There comes a time where you choose to marry somebody. In other words, you say, I'm going to be with them for the rest of my life. And you can have that same thought process. It's too hard. Or you can decide to fight for something that matters. So I want us to work as we go through this, okay? I understand it's difficulty. I understand it's not funness. I get all that. But that ain't got nothing to do with me, okay, at all. So let's go over passive transport. I'll quickly go through some slides of passive transport beforehand. because I know that we didn't really get a good grasp on that initially. And so let's see if we can do all that here. And to make me smaller and put me way down here. I know y'all are going to miss my beautiful face. Got you on all that. Let's see what happens when I do this number, and I may go away. Hey, I don't. Take a look at that. I tell you, this is just genius. Passive transport. All right, we've been through the majority of this stuff already, okay? So remember, homeostasis, that's the desire to have balance, okay? And especially when it comes to the ideas of small things through a membrane, that balance exists primarily through what we define as diffusion, which we'll see on our next slide, okay? So things can passively transport, and remember, all of us should know the definition of passive transport. Connor, what do you got for passive transport? Yes, that's exactly right. So does not require energy. Good work. Nice. I had a way to stay with it, Connor. If this is fifth or sixth period, then well, whichever class you're in, as a matter of fact. I don't even know what class you're in, Connor. But hopefully you got it. So that's called passive transport. So that is the process of moving tiny things in and out of a membrane, and typically they want to be balanced. They want equal numbers. You can see that here, right? As we look at equal numbers of things, we took, let's say, oxygen that was all on the outside in the extracellular space, and they slowly move through, probably slowly, I say quickly, probably for oxygen, down into the cell to make a balance. Homeostasis, small molecules, think of carbon dioxide, oxygen, water, freely able to move in and out. Things that have charges are not very good at that. Negatives and positives, not very good at wanting to move through. Something that we'll see later on, and we'll use some things to make that be better as far as proteins and passive or active transports, etc. But just think homeostasis, movement of molecules easily, or balance, passive transport, moving of molecules easily without using energy, and especially small things that are there. All right, remember, we talked about diffusion, okay? So diffusion is nothing more than molecules moving from a high concentration to a low concentration. It's written right here. It's a definition that we just need to know. The difference in the concentration of the molecules across membrane is called a concentration gradient. So what we mean by that is that we say the difference between this having all of it and this having none of it is very simply what we define as a concentration gradient. We'd give it a value based on that, and then we'd say that it needs to move down a concentration gradient for diffusion, or against a concentration gradient for active transport, several things along that line. Remember, down is like downhill or downriver. Requires no energy. It's the way it should go. It's easy to do. Doesn't take any effort if you think of it that way. Against a concentration gradient is like against the grain. It takes more effort. Got to paddle upriver. However you want to think of it that way. So again, don't overthink the ideas here of diffusion. It should be pretty simplistic as far as that part's concerned. So again, that's called a concentration gradient. The desire of those small particles to move themselves in is called kinetic energy. In other words, it is their own energy that drives their movement in, their desire to be in a place where they're balanced. It has nothing to do with us having to expend ATP or energy from a cell, hence why, again, it's called passive transport. Okay, so those are your three definitions for that. Remember, we'll get to another form of diffusion called what? That's right, osmosis. Which is the movement of water, just the same way as any other diffusion. It's just water itself, okay? All right, so remember the rate of diffusion. We can improve the rate of diffusion by doing pretty much three things. Number one, we can increase the temperature. Hotter things, molecules move faster, molecules want to move over, okay? Size, smaller things, molecules want to move quicker, can go through a membrane very, very quickly, and they want to move over, okay? And lastly, when it says molecule type, we define that as the idea pretty much of things that have a charge. We would prefer them to be neutral. If they're neutral, they have a tendency to move quicker. If they are positive or negative, they have a tendency to move slower. Okay? So I can ask you on a test all sorts of things. I can say which of these molecules would move quicker through a membrane. And I could give you H2O, C6H12O6, and H2 positive. What's the answer? Miss McGee. Yes, that's right. It's H2O. She wasn't paying attention this time, right? Hopefully she was. But H2O, why? Well, because it doesn't have a charge. It is polar, okay, from that standpoint, but has a positive and a negative, so it balances itself out. But it's small. It lacks the charge of the H2 positive, and it's nowhere near as big as C6H12O6. So that's basically all that it takes. I don't think that I can get myself moved any smaller there, so I may go up here for a second just to see. So let's see what we got. All right, diffusion always occurs down a concentration gradient. Will continue until the concentration of molecules is the same on both sides. Of course, as it winds up the same, we call that an equilibrium. Okay, so very straightforward. You would think that you could make this video smaller, but you can't. Okay, not possible. All right, so I'll have to move my face around numerous times for you to continue to get an idea of where I'm at. See, I can do like this, and you have to keep up with me. There I am. That's exactly right. Nice job. Great work, everybody. All right, that's equilibrium. All right, so now osmosis, right? Osmosis is literally water diffusing across a membrane. That's all that it is. It's still diffusion. If I were to ask you that all diffusion is osmosis, that's not true. All osmosis is diffusion. It is true. Okay? Notice this, though. Water moves from areas of low solute concentration to high areas of solute concentration. All right, so here we go. Solutes. What are solutes? We talked about solutes and solvents. Okay? Wes, what you got? Let's think about the idea of a solute and solvent. A solute is what? Great job, Wes. Absolutely. It is indeed going to be the thing that is being dissolved inside of a solvent. So let's use salt as an example. So salt is a solute, and it has a high concentration inside and outside. Let's just think of it that way. I want us to get a grasp on what water is going to do. It's easier for water to move in and out of a cell than it is for salt to move in and out of a cell. Okay, so what I'm going to do shortly is move myself for just a second so that we can see. So I want you to get an idea, if you can, of exactly what I'm talking about. All right? So we have got a cell like this. We have a 2% salt solution. We have got a 1% salt solution. So when they say that water diffuses, or through osmosis, that it moves towards from an area of low to high solute concentration, what it means is that it's moving itself not like diffusion does, where we think about the ideas of it going from high to low. It technically is reversed, but it's not, because it's all about the solute. It has nothing to do with water, right? It's not saying, hey, guess what? There's more or less water out here. That's not what it's saying. It's saying that there's too much salt inside the cell versus outside the cell. So if I needed to balance these things, I needed them to be 1.5 each. That's what equal numbers would be, okay, to create equilibrium. If I needed to balance these two things, I have one way to do that. That is, move 0.5% of the salt over here. Would that work? Yes or no? All right, it would. It would. Would I have to expend energy to move this 1.5% out here? 100%. Would have no choice. I'd have to move the salt out, and if I move the salt out, it would cost me energy. The cell does not want to expend energy. Again, remember, I told y'all, this is nothing different than what me and you are. Okay? Our cells are just like we are. They don't want to create energy or put off energy if they don't have to. We're lazy people. I mean, we're lazy people because our cells are lazy. I don't know what else to tell you, but they don't want to do this. There's another way to fix this problem. We can fix it with water, and water can freely move in and out. So how do we fix that problem? Well, in this case, it's very simplistic. All we have to do is move more water into the higher concentration of salt. And when I do that, I can lower the percentage. In other words, this sweet tea is too sweet. If I add water to it, guess what? It gets less sweet. Kool-Aid. I mix 2 packages in the 1 gallon instead of 1. Well, if I take that 1 gallon pitcher and pour it into another container that's 2 gallons big, and I add another gallon of water, guess what? I've now diluted it, and so it's no longer a 200% solution of Kool-Aid. It's 100%, like it should have been to begin with. So when we say that water diffuses from areas of low solute concentration to high, we move water from here to here. as we move water from here, this number goes up because basically we're taking away the dilution, right? So think of it as having salt water and you taking salt water and pulling the water out. What are you left with? Salt. So this number goes up. As I add water to this, what happens to it? This number goes down. So diffusion, right? Or what we define as osmosis with water, okay? This osmosis is the process of moving water from a low solute to a high solute, not from a low area of concentration to a high area of concentration unless you put the word solute with it. So when you think osmosis, think solute. And technically think backwards from high to low, because instead it's really from low to high, okay? That should not be confusing at all. If it is, please watch this video 500 more times. I don't know what else to say. All we're trying to do is dilute the high one so that it can be lower, take water away from the low one so that it can be higher, so that we can get these two numbers more the same. They still want to be in equilibrium, but we want to do it with water because it's easy. It doesn't require anything. It's passive transport, okay? It's the way to go. It's the easier solution. So just think about that. Keep that in the front of your mind as you're going through all that. I think we'll do much better, okay? So, oh, look, I can get half my face off of there and then get me more out of the way. Nice. Okay, so that's osmosis, all right? All right, so let's go on to our next slide. All right, so hypertonic and hypotonic and isotonic. Everybody watch. All I can tell you is that you're going to get highly confused if you don't follow with me, and you're going to get highly confused if you don't understand the concept we just talked about, but more importantly, if you don't realize all you got to do is take what we just learned and apply that to 3 words. All right? So, hypertonic, here's the definition. A solution in which there's a higher concentration of solute, again, all solute, outside the cell. Okay? So, remove me again. Hypertonic. Higher concentration outside the cell. Six percent, 2%. Still diffusion, still osmosis, nothing any different. Which way is the water going to go, Miss Elena, who's probably asleep? Water's going to go from where? From small solute to high solute. So in this case, where? All right, so from inside to outside. Let me ask a question. If I suck water out of this cell, what's this cell going to do? It's going to shrink. Pretty self-explanatory. I mean, take a water balloon and take water out of it. What happens to it? It shrinks. So, in the case of a hypertonic, more solute outside, animal cells do what? Shrink. Okay? In the case of a hypotonic, which you could imagine, if this is hyper, what do you think hypo is? Hypo is going to be the opposite. Okay? Water is going to be brought in. What's the cell going to do? Swell to the point of almost wanting to explode, and can explode. In other words, they'll bust. Because why? Well, because in that case, it was 2 here and 6 here. It has to move from low to high solute concentration. More water comes in a water balloon. The water balloon eventually explodes. Pretty simple, right? Not really a complicated thought process. So don't miss this, okay? Hypertonic is drawn for you here, and it creates this with animal cells. Animal cells shrink. Hypotonic is going to be the opposite. In other words, we have a 6 here and a 2 here, whatever it is. There's more on the inside of the cell. And because of that, what happens? Water is going to be brought in, and since water is brought in, the cell is going to swell. Isotonic, right, means everything is comfortable. You don't happen to know anything about isotoners because you were far too young for that, but they used to be the glove, especially for all the ladies, right? Isotoners. And if you happen to have ever watched Ace Ventura: Pet Detective, you will know very well what I'm talking about. but pretty much he's doing a commercial, Dan Marino is, for isotoner gloves. Isotoner, iso, right, basically means equal, happy, right, content is what it would mean in the sense of your hands and your gloves, but it means equal. So in an isotonic solution, what do we have? Well, we have equal numbers that are there. I'll show you that on these next few slides, but I want you to get a grasp and an idea, if we can, on hypo, hyper, and iso. Hypertonic, more on the outside. Okay? Water leaves, cell shrinks. Hypotonic, more on the inside. Water goes in. Because water goes in, cell swells, can die this way, can technically die this way. These can explode, these can shrivel to the point of having no water and almost want to die themselves. Isotonic, everything's equal on the inside and outside. Now, just a general question for the future, which we'll see in a second. What do you think about an animal cell? We're not talking plants yet. We're going to get there in a second. An animal cell. What do you think it would primarily want? Would it want to be in a hyper, a hypo, or an iso? That's right, an iso. An animal cell would prefer to be in a situation where everything is balanced. Solute inside and outside is the same. Water can freely move back and forth. It's not causing the cell to swell and explode. It's not causing it to shrink up. Okay? We're going to learn quickly that there is a difference for a plant cell. What does plants need? They need structure. What do plants also need? Lots and lots of what? Water. What's this one? Well, we'll see that, right? As we think about hypos and water being brought in. It creates something in cells that we define as turgor pressure. The more water we can get in it, the tighter that cell can be. The tighter that cell is, the more that plant stands up. Remember, plants don't have skeletal systems, right? They have a cell wall, and they have turgor pressure. Every cell is built on the next cell. That's literally like putting Legos on top of each other, okay? Versus spaghetti noodles on top of each other. It needs some structure. And so we'll wind up seeing that a hypotonic solution, right, happens to do that. These are hypers again. Causes it to shrink, okay? These are hypos. Causing it to swell, okay? The way that I remember this is that hypo sounds like a hippo. So hippos are not skinny, if you didn't know. So they wind up being like this, okay? I'll show you that on these next slides, okay? So that you can kind of see that general idea, but I just want to make sure that we're all pretty much on the same page. All right, so hypertonic, higher concentration of solute outside the cell. In a hypertonic, water will diffuse out of the cell until its equilibrium is reached. Look at these cells. They've all shrunk. Cells will shrink and die if too much water is lost in a process known as, notice this, crenation. You have to know the statement, okay? This is not cremation. We're not burning a body, okay, to turn it into ashes. This is called crenation. So crenation is cells shrinking in a hypertonic solution and dying. If they die in plant cells, we call this process plasmolysis. So crenation, animal cells, plasmolysis, plant cells, if they shrink. So notice, do they still shrink for plants if they are in a hypertonic? Yes. Water still leaves the cell. Since water still leaves the cell, you have no choice whatsoever but to realize that as it leaves, it is a situation of losing enough water, which is awful for plants, losing enough water where the cell's going to shrink. It's kind of like losing water out of a water balloon, okay? So that is a hypertonic solution. You can see these pictures. If you can get this, look, by the way, you're going to draw these for me on a test. Hyper, iso, and hypo. In other words, I'm going to say point blank, oh, you have a hypertonic solution. Draw what the cells are going to look like, right? Draw water leaving them and them shrinking. Isotonic, they're staying the same and they're happy. Hypotonic, they're swelling and they're blowing up. It's not hard. You don't have to give me this in some kind of graphic detail. But my point is that I want you to understand, if you don't get this grasp of water moving from low to high solute, none of this is going to make any sense. And you're going to be lost the entire time. So you got to work on staying focused on that part. Okay, so there's hypertonic. Let's look now, if you will, and let's look at our hypotonics. Very simple. Hypotonic solution in which there's a higher concentration where? Inside the cell. Okay, so what's going to happen? Water's going to move from low to high solute. So what's it going to do? It's going to move into the cell. Very simple. As it moves into the cell, what's going to happen? Well, we're going to have more and more and more water. Are we not? Absolutely. However, they can also take on too much water. Notice this, and notice how we don't have a statement here for plant cells. Animal cells will burst, or what we call lyse. Notice that word, make a note, highlight it, if they take in too much water in a process called cytolysis. So this is cytolysis, and this is the busting of a cell because it's taken in too much water, right? The actual busting, right, the dying of it is called lysing, but we call cytolysis, meaning the lysing of a cyto, which cyte means cell, okay? So the bursting of a cell. So that is a hypotonic solution. Now, plants don't have that issue, okay? I'll show you why in just a second. Isotonic solution, equal. Inside and outside, okay? And because they're equal, basically no issues whatsoever. Water can kind of freely move in and out. Everything is perfectly fine. Everything is happy. Not any major issue whatsoever. Water's pretty much going to move in at an equal rate. So that's just an isotonic solution, okay? Not going to go over that slide for any length of time because by now, if you know one of them is too much on the outside and one of them is too much on the inside, you know that the next one's just equal, okay? Should not be very complicated, okay? So, cellular solutions, right? Let's take a look. Plant cells, prefer what? Well, hypotonic. Why? Well, because it sucks water in. And plants have this big, huge central vacuole that helps to store water and pretty much is what it's going to use to then be able to generate all of its energy through photosynthesis, right? So, since it needs that water and that water helps to create both its energy as well as its structure, it sucks as much in as it can. So look at this. Plants prefer hypotonic solutions, and as they take it in, they create what's called turgor pressure. Because they have a solid cell wall, they are nowhere near as prone to exploding. Okay? So they will bring in lots and lots and lots of water. That pressure will increase, creating turgor pressure. The cell now has a cell wall and turgor pressure to give it some structure, and so now that cell is pretty rigid. Okay? Turgor pressure helps plants keep their shape. Notice we could obviously understand for an animal cell, what do you think it wants to have? Well, it very much wants to be what? It wants to be in an isotonic solution. Okay? So you can see here, right, this word of flaccid, right, we use to talk about the idea of something being limp, something not being strong, right, and rigid. And so here, this is exactly what happens in an isotonic solution for a plant. It doesn't have that turgor pressure. It does not like isotonic solutions. It hates hypertonic because in that case it's losing all of its water and it needs all that water, right? In this case, typically it's going to die, right? Plasmolysis is what will happen again. Remember, that's plants losing too much water and dying, okay? So everybody's got that? Hopefully not anything overly complicated there. All right, so facilitated diffusion. It occurs when carrier proteins embedded in a cell membrane help move solutes such as glucose across the membrane, okay? So this is passive transport, again, because why? It does not require energy, right? So we can do this with channel proteins. You can see those here, right? Carrier proteins, which basically are made for specific things to fit in them, and they pretty much open and close like a gate, okay? Known as facilitated diffusion. Primarily, its purpose is glucose. Anybody want to go on a limb and explain to me why we would have a passive means for getting glucose into a cell? That is right, everyone, because we need glucose nonstop to break it down to create ATP. And if it requires us a boatload of energy to bring in before we then have the boatload of energy that we make, our total boatload of energy will be nil and zero. We don't want that. So instead, what I want us to see is I want us to get this concept that we are having the ability to get an energy maker into the cell with almost no work and effort, which is great. So that means then we can freely expend that energy to make more energy. So this is called facilitated diffusion, primarily glucose, okay? It's not the only thing, but primarily we're moving glucose across the membrane. So I want you to kind of put those things together. Facilitated diffusion. Notice it can happen here with protein channels, or what we call channel proteins, or as well what we call carrier proteins, okay? All right, so there is the end of that presentation. There is no need to put your stuff up at all. So just pump the brakes, settle down. All right, so let's take a look at our active transport. Welcome back. Isn't that fun? Okay, so if passive transport does not require energy, active transport requires energy. All right, so this is a sodium potassium pump. I showed you this, as a matter of fact, when I had my foam pieces the other day that I was showing you, talking about the ideas of proteins being used to move things in and out of membrane. That was a sodium potassium pump, primarily because in it is a 2 in 3 out situation, which we'll get to. But a sodium potassium pump is a very prominent thing. Why would I need potassium at all? Let's see if we can take a look at this picture and figure this out. So take a look at this right here, and let's see if Bryce can figure this one out. Bryce, why would I need more potassium, Bryce? That's right, Bryce, because this ATP is going to have a broken, right? We're going to break it to create energy. That's what we do. We take the phosphate off, and I need more potassium, right, to create more phosphate, which you can see here as we bring it back to put the ADP back to ATP. So we need lots of it in and out of a cell, okay, to do that. And so to do this, it requires energy as well, so we have to expend some energy to make this happen. In this case, primarily, it's because we're bringing other stuff in, okay, as we do this. And so you can kind of see, and we'll go over this idea of the sodium potassium pump specifically, but what I want you to gain a concept of is that we are actively spending energy, okay, to get things in and out, because normally it's because these things, look at this, right? Here's your sodium, potassium, they're all positive. They don't want to move through the membrane on their own. And so we have to help them do that, right? Active transport utilizes ATP to move materials against a concentration gradient. What does that mean? Against a concentration, that means areas from what? Right, from instead low to high to low, okay? From an area of lower concentration to higher concentration. That's active transport. Just get that in your head right now. In other words, what we're doing is we're taking things that we need and we're moving them from low areas into places where we may already have more. We already got ATP everywhere, but we need more phosphate to help build it. And we need more this. So we have to pump it into us. We have to spend some energy to get some energy is what we're doing. The process that moves sodium and potassium ions through a cell membrane is called the sodium potassium pump. We'll go over it in detail in the sense of this 3 out, 2 in thing here shortly. Okay? So everybody's hopefully on the same page with me there. Don't know if I just double moved us. Let's see. No, I did not. Okay. There we go. All right, so membrane potential. The sodium potassium pump moves three sodiums out for every 2 potassiums that move in. It's a question on your test. There's nothing you can do about it. You can see them here, right? Right? Here is our sodiums. There's 2. We're about to add a third one right there. And then we will be able to beep, beep, beep. All 3 of these will go out, okay? We will then be able to bring in only 2 potassiums. You can see them come in here, okay, as we try to recuperate our phosphate that's right here, okay, that we had to burn off this ATP before. These ions are exchanged. Charges within the outside of the cell are changed. The difference in charge is called a membrane potential. So the difference in this positive and down here, this other positive, the difference between these 2 things is called a membrane potential. Okay? All you can do is just memorize the statement. It's just as simple as that. Know there are, again, 3 sodiums out for every 2 potassiums that come in. Know that it requires energy. Know that it's called active transport because of that. Okay? And very much this will be involved in lots and lots of things for our cell. They're very, very vital for us, okay, as far as continuing our energy production for each cell. Okay, everybody's good there? Fantastic. I'm trying not to go too fast. If I did, somebody go back and pause the video. All right, so let's look at endocytosis, pinocytosis, exocytosis, phagocytosis, all the above, okay? So the movement of large complex molecules across a cell membrane is called bulk transport. Large molecules, food, right? Things it needs to eat, whatever it happens to be, okay? Water, you name it, all right? All of those things are called bulk transport. They're going to enter into vesicles. Remember, a vesicle is nothing more than a package. Remember, the ER can do it. The smooth and rough ER can do it as they send it to the Golgi apparatus. The Golgi apparatus can make vesicles as they're sent out as well. So nothing overtly complicated about that concept. But what I want you to understand is endo means, do we all remember perhaps studying our Latin? Into. So this means we're bringing things in. Okay? So if you happen to look here, we pretty much reach up, right, in this case, in phagocytosis, and we get solid things. The membranes will come together. It will pinch itself off, and it'll create this, known as a phagosome, right? It's nothing more than a food vacuole in this case, which is still a vesicle. Okay? Look here. We did this with water, pinocytosis. Okay? Same exact thing. Right? Here's our receptor-mediated endocytosis. Okay, in other words, that we did this because we had a protein coat and it told us what we needed to bring in primarily and how we needed to bring it in. And we had a receptor that actually bound to things to help hold them in. You can see here, here, and here. And it basically won't do it until the stars align, if you think of it that way. So they actually come in and are collected by these receptors, and that tells it then it needs to close itself in. Okay, that is receptor-mediated endocytosis. All right, so again, don't be confused by anything. This is not overly complicated. Bulk transport is moving large molecules into the cell. We need to do that for all sorts of reasons. All right. Vesicles are the things that are created once we do that, the packaging of whatever we brought in. Okay. Typically, what would happen is, like in the case of a phagosome, right here, is that a lysosome, which dissolves, right? Remember, it's a recycling center. It would actually come and join this membrane. And then either this would move into the lysosome, or the lysosome would let all of its enzymes out to go into here. It would stay within that circle, right? If I can get my camera right. It would stay within that circle, and then it would eat and dissolve what's in there. That way it could be used for energy and those types of things, brought to other parts of the cell, those types of things, okay? So again, endocytosis, bringing things in. Remember, you're going to have to know these other words. We'll get that in a second, but keep that at the forefront of your mind, okay? So phagocytosis, right, it's just what I just told you, food particles in. That's all it says. You can see it right here, right? Process of a pseudopod extending around a food particle. Here's your pseudopods, right? Pseudo, fake is what that means, okay? Pod means an arm. It's the fake arm. So it's saying it reaches around, and it creates this as it comes inside, okay? That way, guess what? When this part here joins this part here, it will be able to do what? Stay as an intact membrane. Can the membrane of a cell just open up and be done? Nope, it cannot. So again, phagocytosis, it describes pseudopods extending around a food particle. Once around by the pseudopod, the particle is enclosed in a membrane pouch forming a vesicle. Not anything to do besides memorize and put it in your head. If I were to say on the test, phagocytosis is a form of exocytoses, you would tell me no, because it's a form of endocytoses, right? It's bringing things in. Phagocytoses is the process of bringing water into a cell. Evan, false. It's not. It's a process of bringing food into the cell. Okay? So again, get a concept there that is hopefully simplistic to understand, which is it's not hard to get endo and their groups and then exo and their groups and just to keep them together. And if you do that, then you won't struggle. If you look at this the night before the test, you're in trouble. Okay, so don't be that guy. All right, just don't be that guy. All right, so again, phagocytosis, bringing food in. As it brings it in, it creates a vesicle. Okay, very simple. All right, pinocytosis or phagocytosis, let's continue this again, sorry, is known as the feeding process of a cell. Feeding, why? What's it bringing in? Food, okay? Again, vessels broken down by lysosomes, you can see that here, okay, releasing their contents into a cell. It's the feeding process. What do you think the drinking process is of a cell if phagocytosis is the feeding process? Well, there you go. Pinocytosis, and you'll see that here in just a second, okay? I'll give you just a second to write that down, since it looks like my time is getting real close. Okay. Pinocytosis, the drinking process of a cell, It's a type of endocytosis that describes the process of a pseudopod extending around fluid droplets. Nothing for you to know, but just to know it, right? Pinot is an endocytosis, the bringing in of something into the cell. Specifically, it's the bringing in of water, right? Or anything that's on the outside that's fluid that it needs. It's the drinking process of a cell. Still a vesicle, right? That's there to do its thing. Keep giving you a little bit of time here so that you can write things down, because there's not much else to say about this concept. Once you know it, and it's just move along to the next thing. All right, so if endocytosis is bringing things in, exocytosis is what? Moving things out, okay? The movement of things out. Proteins made by the ribosomes are packaged into transport vesicles by the Golgi apparatus. They are, we know that, okay? They're then sent where? To the membrane. Transport vesicles fuse with the cell membrane as proteins are secreted out of the cell. So simply, notice here, right? Here's Golgi apparatus. It's packaged this. It's brought this to the edge of the cell. Once it is bound to the cell, it will open up. It will release these three things that are here, right? This is liquid, let's say. Same thing here, right? Lets these things out, okay, as it bonds. Now, think about which one of these processes, endo or exo, creates a larger membrane. That's right, it's exo. Because what happened? We just added this vesicle membrane to this membrane, and it grew. The other way, we're pinching off our own cell, and we're losing in endocytosis. Okay, so if I were to ask you in the test the difference in those two things, we have a concept of that, okay, hopefully. So again, exocytosis, moving things out. Golgi apparatus ships, transport vesicles fuse. So in other words, these vesicles are nothing but transport vesicles, what it's telling you. They fuse with the membrane. Whatever's inside of it goes outside the cell. All right, very simple, okay? All right, hopefully everybody's good. We didn't have too many problems today. Everybody did well, and no one got written up, and we learned some stuff. Tomorrow you have a review for me. On Wednesday you've got work for me to do. On Thursday you have a test, and on Friday we have more stuff to do. So let's be prepared for all of that. I look forward to being back with you whenever I am. Hopefully you'll be back before next week. If not, I'll be back next week. And so we'll kind of go from there. So look forward to seeing you guys. Hopefully everybody did great today. If you have any questions, feel free to rewatch this video. It's about as simple as I can say that. Okay? Thank you so much.